So let's begin with some chemistry of oxygen. So that is also related to important topic for chemical biology. So chemistry of oxygen. So let's talk about some Lewis electron structure of oxygen. So that we have oxygen here, the atoms and oxygen atoms here, and then the electrons of oxygen is actually 6 electrons here. So this also 1, 2, 3, 4, 5, 6 electrons. Then as you know, that remember your chemistry 101 class, that is equal to oxygen double bonds to the oxygen. So this is generally accepted Lewis structures of oxygen. But in some cases in actual biological classes, it's also acceptable to say that you can have 1, 2, 3 here, 4, 5, and 6 electrons of oxygen, and also 1, 2, 3, 4, 5, 6 oxygens here. So that is, of course, it's not a appropriate model for the Lewis structures, but actually this is happening for explaining the chemistry of oxygen in biological systems. Actually, so we have, say, when you have four electrons, for example, 4 electrons comes, then that becomes H2O of 2 molecules, because you can have, say, 2 electrons coming, then 2 electrons is coming, then these atoms can be H2O here, and then 2 electrons per here, that can be H2O. So two molecules of H2O here. But when you have less than 4 electrons obtained, that we call that is reactive oxygen. We called reactive oxygen species. So we called ROS for this. So the reactive oxygen species, the cases that we have the first electron, for example, say, so you have, I will continue this here, and then when we have the first electrons actually, then we have, say, this one is superoxide anion, we call it. This is the first electron. So this is the 1 electrons comes. So we have 2 electrons comes. Then we have the so back to the blue, and then we have in here, and then we have now first and second electrons is coming. So this is the peroxide. This is peroxide. This is the 2 electrons obtained. And the 3 electrons here is the hydroxyl radicals. So we have OH radicals here. This is hydroxyl radicals, which is 3 electrons. So the less than 4 electrons obtained for the oxygen, we call the reactive oxygen species. That is now hydroxyl radicals. The superoxide anion, peroxide, and hydroxyl radicals we call. So these are generally reactive. Chemically reactive. So that means they can attack to existing biological molecules such as DNA and RNA and proteins. So they produce the ROS, then attack, and they then give damage of these proteins in DNA and RNA molecules. In particular, so we have some the special system that you have some pathological conditions. So the pathological conditions means that we have invaders. As the viruses, for example, this will be the linkage between the coronavirus lectures and this lecture. So this is a background of lecture. So there are also bacterial infections. So we have some the cells we called macrophage and neutrophil, neutrophils. Then neutrophils. Then when the bacteria or virus, for example, so when the virus, for example, comes here, they infect it. Then the macrophage, the role of macrophage is actually this is the immune cell. So when our non-self molecules such as virus and bacteria comes into our body, then the immune system, the first immune system, which is macrophage, that recognize, oh, this is not our cells, so that they recognize a foreign cell, then they attack and encapsulate, then making the inside of these particles go inside, so that we have the macrophage. So I'll draw the red one for, say, for example, this coronavirus, for example, or other viruses or other bacterial cells can be also same cases. So they envelop, so they engulf. We call the engulf of these foreign particles into the cell. So they basically, this macrophage remove these foreign particles. So this called phagocytosis. So this is phagosome in here. So in these structures, then they produce, and the macrophages alert other cells that I am having the foreign particles. So they actually produce the ROS, which is the reactive species of the oxygen that produce to the environment, so that they can be other systems, other cells are alerted by this reactive oxygen species. In more detail, that you have, say, in here you have the phagosome that has you have NADPH. Oxidase exists. So the oxidase means they're taking all the electrons, or providing the electrons here by the reactions of the oxidase. Then it means you have oxygen, for example. You have oxygen, then providing oxygen to this existing oxygen molecules to become, in this case, is that, for example, superoxide anion. Superoxide anion by the action of the NADPH oxidase, which is existing to the phagocytosis, phagosome process. Okay. So that means all the process that in the pathological conditions that our foreign particles comes to our body, and the immune system, the cell macrophages, and engulf those foreign particles, and basically at the end of this reaction they produce reactive oxygen species. So that becomes alerting the neighborhood to accelerate this process, because it's not a single particle has come. Actually, a lot of particles has come, numerous particles come. Then they have to activate other cells to recruit and attack together. Okay, so we talk about the chemistry of oxygen. So the second terminology will be translations. So translation means that we have, say, DNA becomes RNA. This is what we call the central dogma, and then becomes a protein. Then this process, the RNA becomes a protein. So this is a coding. This has the coding for making the protein. So this all the process related to a biochemical process we call translations. But that is a translations. So I will briefly remind of some terminology. And then also RNA dependent and RNA polymerase. So as the name indicates, so you have RNA backbone here, and then they make actually duplicated RNA. Basically, this enzyme producing a lot of different RNA structures. So you have AU, GC, then you have this RNA polymerase comes in here, and they are in the cell, they have a lot of nucleotide. They incorporated UA, GC, CG, and they have another strand they make. So they can basically amplify the number of the RNA transcript, which is the enzyme that is responsible for this process. And also the tumor necrosis factor alpha. So we call briefly TNF alpha. Basically, this is protein. This is a protein that is secreted. Is secreted by a macrophage. So that's why this is related. So the macrophage, remember the previous one that I mentioned about the reactive oxygen species in the macrophage engulf, taking up the foreign particles, they produce reactive oxygen species. Also, the very, very important protein this macrophage release to the cell or environment is this tumor necrosis factor alpha, TNF-alpha. So they also play a lot of the subsequent process for the inflammation process. Okay, so now, so based on this background, so that we can continue our real topic of the coronavirus. So this coronavirus that So let me just begin with some easy topic. So how we can get infections by the coronavirus. As we know, I think everyone knows it, but just briefly remind that this is fecal source. So the fecal means you have our body have some secretions, right? So the fecal we have known that this coronavirus and influenza virus is existing actually in the fecal in here, and also some aerosol system. So the aerosols typically we know that this is existing for about 3 hours, and also the surface. Surface is about 24 hours. So that the infected person by coronavirus, so by accidentally this hand and touch their his face, and then also the nose, because we don't see the virus, and touch the surface, and then the virus can be existing on surfaces here for actively for 24 hours. Also, we have some breathing process. We provide aerosol. The aerosol has the virus from the nose, and then some studies have shown that the aerosols by the coughing or something like that. So you have some floating around with attached to dust or other. So we have a lot of carbons and particles here. We cannot see it, but actually we see when sunlight come in the morning, then you see a lot of particles in here. So they attach to that particles, and they existed in the same place. So that when other people through this area and also breathing that area, then for 3 hours it's possible to be infected. So that is about 3 sources of the infection. Okay. So when you think about when you breathe by accident, surface or breathing of the area, contaminated area, also the surfaces you can touch, and then by accident that hands contaminate your hands and then touch your faces and breathing. So then directly the coronavirus goes to the lung. So that's why the problems begin. So I'll explain that the part. So then you have here, so you have, say, draw it this way. This is we call the alveoli. Alveoli. The alveoli is basically lung. This is a basic unit that exchange the gas, CO2 and oxygen. This is a unit of the very, very small. But the number of the alveolus actually existed. It's like a grape. The grape has some main stream, and you have a lot of grapes. It comes in. It's like the alveoli structures look like that way. So the air comes in here. This is air comes in here through this line. So at the end, you have the line of the cell. For example, like this. And I will draw a little different way of the cell. And then cell, I will draw like this way. So this is individual cells. This is a nucleus cell. So this is a pneumocyte we called the pneumocyte. This is a single layer. So the air comes in. This is a cell that directly interacting with the air that we breathe. So it's a contaminated air, which is coronaviruses here. They eventually viruses contacting with these cells. So this is a pneumocyte type 1. This is a majority of the population of this pneumocyte is type 1, which is they actually exchange the gas. Exchange CO2 and oxygen. This is the main role of the pneumocyte 1, type 1. But also you have a pneumocyte type 2. That's why I draw a little different way. Okay, I draw a little different way. You have a different shape of the cell. That is pneumocyte type 2. The main role of the pneumocyte type 2 is they secrete lubricant. The lubricants, you know the lubricants, when we breathe, actually our individual alveolus, they expanded and shrink for many, until we die. Until many times we can do even 1 minute. So for this the existence of this lubricant, actually this friction force becomes nearly zero, so that this alveoli can be freely expanded and shrink because of this lubricant. So when the coronavirus comes in here, then inside now, they actually attack pneumocyte type 2, not 1. So because if I throw this cell in here, so this pneumocyte type 2, for example, in here, at the surface of the pneumocyte, they have a very specialized protein. It's called ACE. So we have some proteins that we called angiotensin 1 converting enzyme. This is a membrane protein. So the membrane proteins are just in membranous. That means it exists on a surface exposed. This is to be called ACE, ACE. These ACE proteins exist in the pneumocyte type 2. The coronavirus, we call this a spike protein. The spike protein recognize, they bind actually. They bind here, in here. And when they bind, the cell is engulfed and making these old structures inside to the cell, so that they make the coronavirus located inside cell by the help of the ACE protein. This is called endocytosis, right? This is, endocytosis means endo is inside, right? Endocyto is a cell. Endocytosis. So the system that the cell has some particles comes and attached to the membrane protein, they encapsulate it. Okay, so this is the endocytosis process, and then eventually they comes out with the RNA, because the coronavirus is an RNA virus. They release their own coronavirus RNA inside the cell, and then they want to amplify by themselves, because the coronavirus itself is not the living things. That means the coronavirus itself cannot amplify. Cells, virus, spectrophoresis cells can divide by themselves. So 1 becomes 2, 2 becomes 4. But the virus is just a particles by itself. So they're not living things. But they become living thing only when they infect the cell. So they becomes alive now because they're inside the cell. This because host cell. And the host cell, this is a ribosome. The ribosome is the area that the RNA binds and make a protein. So this is translation process, as I mentioned before. So now this they make a protein. Proteins here. So this is proteins is actually coronavirus protein. Coronavirus protein. At the same time, as I mentioned before, there's an RNA-dependent RNA polymerase is inside the cell, not themselves. That they have a host actually kindly provide RNA-dependent RNA polymerase. So it's kindly this host cell provide this enzyme. So this coronavirus RNA comes, they make duplicated also. It becomes 4 of the RNA, so they multiply this RNA of the coronavirus. And also, some of the proteins actually, some of the protein that produced by this RNA is actually there's a protease. So protease means that the protease, like a trypsin and papain that cleave the protein itself, right? So that this the proteins that produced by the RNA virus, actually the protease comes, they make a variety of different proteins. So I'll draw it using different colors now, because the protease make this protein providing different types of the protein. So some are black, some are blue, because of the action of the protease that chopped up this protein, for example. Now they produce this. So this protein is basically most proteins are actually coating proteins of the coronavirus. Then this protein encapsulate this RNA, which is also amplified by the RNA-dependent RNA polymerases. And this protein encapsulate this and then taken out. Right? So this encapsulated together, and then they make another new coronavirus with the RNA here. Is existed. So they can, since the RNA-dependent RNA polymerases produce tremendous number of the RNA, also the protein the same, so they can produce a lot of different coronavirus inside the cell. Okay. So these cells then kill whole host, and then they released. So now you have a virus particles outside. But the thing is, think about the back of this. All these happenings are here, and they release the virus here, and then they find another pneumocyte type 2 nearby, and then they do the same thing. So in this alveoli, they basically produce a lot of number of the coronavirus at the end of the process. Okay, so this will be the first part of the coronavirus section. Then we'll continue the rest part, which is part 2 coronavirus section next time. Thank you very much.